You notice that there were 3 items to download. Actually, there's another one. I've added this link. It's called the not so short introduction. Oh no. The not so short introduction to S4. In other words, for S4 classes. So this particular document talks about S4 classes, and it's 60 or 70 pages, I think. But it does it in a much more complete way than the book does. Which brings up a point. I've been trying to decide what to do with S4 classes. And what I did with S3 classes is we did 2 examples in class, and then I assigned one. The 2 examples we did, if you recall, is the t-test and then the len mod. And then I assigned you the one way. Now, the part of the reason is because the book has snippets of stuff, but it doesn't sort of work through an example, and it's kind of hard to put together how you do a complete S4 class to accomplish some task. Or it could be multiple S4 classes, particularly if you have inheritance. And so that was why I did the t-test and the len mod, is because that was sort of a complete example. Well, the situation with the book in dealing with S4 classes is the same and perhaps worse. That it's a good reference if you know the material, but it's not necessarily good to tell you how you build a particular S4 class. So for that reason, and so this particular document, the not so short introduction to S4, is better in that regard. But first of all, I updated slightly, not a lot, the Chapter 3 S4 notes. And I'm not too sure how much time I'm going to spend on those because, again, what I think I would prefer to do is to do 2 examples like I did for the S3 classes. Because I think that gets at the material a lot better. Now, once you sort of know some of the ideas, like what is a generic function in S4 classes, then you can go back and look at the references or read the material in the book, and it will strengthen it. The book gives a lot of detail that if you are doing heavy-duty S4 classes, you may need, but for our purposes here, to introduce you into S4 classes, that's not the case. So, as a result, so again, these are slightly updated, but it's only slightly. So, what I want to do today is I stuck this up here under fall recess, and those notes actually contain a .omd file and the document that this RMD file is based on, which is basically the notes of the particular person who did this, and this was a biostatistician. The SMP stands for SNP, and I wanted to give an example like this because it's a bioinformatics type example. And then, as you recall, Frederick Leisch was the one that did len mod for S3 classes. But he's also done a tutorial for S4 classes. And so that's what I'd like. So if you click on those, they're in zip files, these 2, so you have to unzip them, and then you'll see the .omd file if you want to follow along with me. So first of all, let's go then into RStudio. And let me clear some things. I'm going to clear the workspace just to make sure that some extraneous stuff doesn't stick around. I want to start out first of all with. I want to start out with the snip case. It's the shorter of the two, and perhaps a little bit easier. I have several of these. Yeah, this is it. So I just want to look at this. You can actually look through this whole slideshow. This particular person is actually our speaker today. We do have a speaker at 4 o'clock from the University of Washington in biostat. Got a PhD in statistics from Michigan. He actually overlapped with Mark Culp. He's actually in biostatistics at the University of Washington. So again, I'd like you to try to attend the seminar if you can. The thing I wanted to mention is that the Fred Hutchinson Cancer Center is at the University of Washington. In fact, the speaker's wife works in the Cancer Center. So you might want to read through this whole thing. I'm going to focus on. I'm going to be focusing on starting here, implementing S4 classes. So I'm going to be focusing on this. So I'm going to go over to. Let me just. I'm going to go over to. I'm going to sort of leave this open so I can refer to it if need be. And so. So I'll look here and look at the .omd, and then I'm going to set the working directory to that. Okay. So the objective here, if you would, does anyone know what a SNP is?
Single nucleotide polymorphism.
Yeah, it's a single nucleotide polymorphism, and if you would go in and look that up, even in, say, you would see, for example, in Wikipedia, what you see is that you have a change, a CG to, say, a TA, and basically that's associated with an allele. And this is sort of how we differ, because our genetic code is not all the same. And so we have these differences, but some of these differences, these SNPs, can be related to disease. And so therefore it's become a sort of very intense area of study. And there are a lot of SNP databases out there, these very large databases. Now, so you might want to, just for background reading, you might want to read a little bit about this. And I believe that Wikipedia gets some negative comments in terms of how accurate it is, but this is fairly basic stuff. So if you want to find out a little bit more about the background of this problem, you might want to do some reading. So what I'm going to do is we're going to look at an example from the human genome. We're going to look at just a couple SNPs, but this course could be expanded to many. And in fact, one of the things in the, I believe it's the next chapter, looking at I/O, part of what we want to start doing as we go through, for the rest of the course is some of these external databases, you can pull in material from them. And so in other words, you could pull in the SNP information from these databases. And there are a number of databases that do this. And we have built-in functions in R that can do it. And then we have a lot of built-in stuff within bioconductor packages that can analyze this type of data. But what we're going to do is to actually create a class. And at this, we're not going to do a whole lot about it at this point, but we're going to illustrate this in terms of how you use or how you set up S4 classes. So as you know, set class, we're going to establish a class called SNP locations. So that's the name of the class. And for this, we're going to just have a representation. There are other things that we could have with set class, such as contains. And my other example, I'll be having subclasses, so we'll be looking at contains. We can also have prototypes, which effectively give initial values for these things. For the slots, we define 5 slots, and those are defined in the representation. And it says genome is character. Now, the name of the genome we're going to study is HG19. Does anyone want to guess what that is? What do you think HG stands for? Human genome? And 19 was, I think it's the most recent, dated around 2009 or so. So there was an HG 18 and 17. And so as it iterates, but there's been mapping of the genomes of many species now. And so there are various databases that you can get this information. I don't know to what extent, how far some of the genomes have gone in terms of SNPs, but at any rate. So the genome is simply, in this case, it's simply character. And then the SNP ID is it's a character. These are the SNPs, the list of SNPs of length n, which could be a lot of them. In this case, the example we're going to do, we have 2 of them, but there could be lots and lots because there are many, many, many SNPs within the human genome. And then the chromosome where each SNP is located on. And then the position on the chromosome. So those have all of length n. So if you've got n SNPs, you have n locations. So for the SNP, you know the chromosome and you know the location on a particular chromosome. So we're going to set the class. So that defines the class SNP locations. Now, so how do you establish an instance of the class? There are several ways. The most common way is to use the new function. So you say new, and then you specify what those values might be. But instead of doing that, and there's also an initialize function that you can use, and perhaps that's the more common way. But instead of just having new and specifying all the possible values, we're going to actually have a wrapper function, a constructor function, or a wrapper function that wraps new. And then we're going to, and that wrapper function will have a better name than just new. And so we have a wrapper function, which in fact is called SNP locations. So in other words, SNP locations, and it's okay to have the same name because SNP location above is the name of the class, whereas this is now a function. So they're not the same. Even though the names are the same, they're not the same types of objects. So SNP locations is a function of the 4 slots: the genome, the SNP IDs, and so forth. And the only thing in that function is it calls new with those 4 slots. So the arguments to new, are going to be, the first argument is in fact the class, which is SNP locations. And then we have the 4 slots, genome equals name. So you pass it, you're going to pass it those 4 arguments, and you're constructing a new object of type SNP location, of class SNP location. So you create a new object with those particular slots. And so what's the data? Now, of course, in reality, we would go out to a database and pull in might be thousands or tens of thousands. So N could be quite large, the number of SNPs we pull in. We pull in not only the SNPs, but we pull in which chromosome they're on and where they're located along that chromosome. Okay, so typically what you would do, and as you learn a little bit more, you would go out and suck this information in from a database. And then the idea is that we're building something that does a few things. But in reality, there are S4 classes in bioconductor that do a lot with SNPs. Okay, so let's do the constructive function. And then we're going to actually create an instance called SMP LOCS. That's actually an instance. So it's an object, which is an instance of the class SNP locations. And so notice that the argument's HD19. That's a character string. That has to match. With S4, you have to match. If I say it's character, it better be character. And I did say it's character. When you go back and look at the slot, it says it's character. The next one is going to be a character vector of length n. And so in this case, it's these are actually the SNPs, the SNP IDs. And so those are, in this case, n is 2. Okay. So it's a character vector. And then next are the chromosomes, which is again a character vector. And then these are going to be integers. These are going to be integers which indicate the position. So let's do that next. And so now if I sort of say, whoops, what's going on? What did I do? Not. Let me go back and find that. Okay, that's. What's going on? What now? Is yours working? Did I accidentally? So let me check my parentheses. That's right. Oh. No, that's correct. And that's correct. I'm just checking the parentheses to make sure that this is puzzling. It worked just a little while ago. This is not good. Yours is working?
Yeah. Looks like the same code.
Yeah. As I said, I just went through this code. Is that the rest of you getting it to work? Are some of you doing it? Yes. Why does it think genome is a function? I'll tell you what I'll do. I'm going to not save this. Let me open up. I'm just going to try this. I'm going to try to execute the whole thing. Yeah, see, that worked. Okay, this worked, so why is this not working? I'm going to go to the workspace and get rid of this. Start over again. That works. Are you running it on RStudio, or the ones who, or are you running it on, I mean the server, or are you running it locally?
On the server.
This is weird. I mean, the reason it's weird is it's working here. I mean, this is what the output should be. So it's executing completely, but it's somehow. But I've gone through it step by step before, and it works, so I'm puzzled. Let me just out of curiosity. I'm going to download. This is running it locally. I'm going to launch. I had an old one up there. So I want to make sure I have the new one. Download snip. Yeah, let me. Okay. Actually, I want to check something. Yeah. Let me try it here.
The comma in the integer, the position.
What now?
There's no comma.
It's the last one. So you don't need a comma in the last one. You're talking about the comma like here? You don't need it there because it's the last. It's the last one. At any rate, I'll have to look at why it's not working on RStudio. As far as I know, that's the exact same code. So I'll just run it here. So we've now created an instance called, we just arbitrarily called it SMP LOCS. We named it. So we have an instance. So you could create another instance if you had like HG19 and you had some other SNPs and you had their locations and so forth. You could create multiple instances. So you can create multiple instances. These are our objects, and the class of these objects will be SNP locations. Is everyone clear on that? Now, of course, setting up a class isn't much good unless you operate somehow on the data. The data is contained in the slots of this instance. So we want to be able to define methods on this instance, or on all instances. It'll be the same methods. So the first one, let's just get the length, which will just record how many of these locations we have. And so if we run this, then this defines a method length. It says set method, so we have a method length. And then the second argument is what's called the signature. The signature is, in this case, it's the class. So it's a method associated with this particular class called snip locations. And then we have a function. In this case, the only thing the function is the length of the number of snip IDs. Now, this notation, x, is the argument to the function. And so it's going to be passed an object that's of type class snip locations. So x will be the particular instance that we've created or that you apply this method to. And we apply it as a function. This is sort of a unique thing to R in some ways because it's combining a functional programming language with a class-based programming language. And not only that, you can do it interactively. In other words, I in effect am defining this class interactively. And so this is unusual. If you program in Java or something, this is not the way it works. Notice the x at SMPID. What the at is an accessor. You have the object or the instance you've created at and the name of a slot, and it extracts it or it accesses that slot. So that's one way you can get the slot value. So that actually returns x, which is the name of the object. That's the function argument. But I'm going to be passing it that function. What I'm going to be passing to it is, in fact, the name of an instance of the particular class snip locations. And so it's extracting the slot value, and then it's going to take the length of that. Okay, so that's what we're doing with set method. And then to test it out, we just say length, and then with the snip locations is the name of the instance that we created. And so let's see what happens when I execute this. What should it be? Should be 2, right? Because we just had 2 snips. Or 2 snip. It was a vector of length 2. In other words, n back where I had the set class, it says it was a character vector of length n. In our case, n was 2. When we constructed it right here, there were 2. Okay, so now what we're going to do is define some generic functions. And in order to define a generic function, you need to do this. So in other words, the way we define length, we didn't define it as a generic function. In order to define something as a generic function, and a generic function is, if you remember in S3 class, it dispatches based upon what the classes of the arguments are. And unlike S3 classes, you can have multiple arguments you can dispatch on. And we'll see an example of that later. So I'm going to define the genome as a generic function. And I'm wondering if this is something that I had done previously, and I thought I had gotten rid of all that stuff. But here I do define genome as a function. It's a function, first of all, I'm just defining it generically. And then I'm going to set method for genome. And I'm simply, the only thing it's going to do, it's what we call slot getters. In other words, it's going to get the values of the slot. So I'm defining a generic function that will pull out the genome. So in this case, if I say run, I now have a method. And so I can say genome, and I can have the object, and it tells me what it is. Instead of using the @ sign, which is another way of doing it, but generally speaking, we like to have more descriptive functions for doing it. So I'm going to do the same thing for snip ID, and I'm going to set its method. So snip ID becomes a generic function. Chrome for chromosome also becomes a generic function. And likewise, the position is going to be a generic function. So let's test it out. So genome is now a function. So at this point, genome did become a function. And what I'm wondering is the last time I did it is somehow I thought I erased that R workspace. And maybe not. Because at this point, what that arrow was saying was genome. It wasn't able to find the function. So something may have been going on relating to the fact we've now defined genome as a function as well as a slot. So it's a slot value, but it's also a function. So if I say genome of, and then now the argument is the SNP locations that I created, right? So SNP locs is the name of a specific instance of the class. I apply these generics to it. Let's see what happens when we apply genome to it. So what it's going to do, it's going to give you HD19. And when I apply it to the locations, it gives me the SNP IDs, I mean, it gives the IDs for those two SNPs. Okay? So we created some generic functions. Now, you remember if S3 classes, you had a print method. Here we're going to have a show method. Show is sort of like the print. And so if you print an object in the S3 system, it has a print method. If you on the command line, if you put an object, there's a default print method called for that object. But if I now put like SNP locations, which is an instance of a particular class, it will now call this show method to display information about it. Now we're not this. So what? Let me go ahead and execute this, and then I'll tell you what we did. So show is the method, and it's associated with the class SNP locations. So the signature is SNP locations. And then I have a function, and the object, I'm going to pass in an object of types SNP locations, and I'm simply going to print out a text message. And so it says cat. That means concatenate these character strings. And the class object, in other words, if I say the class, so if I would say class of SNP-LOCS, is that what I did? So it's SNP locations is the class, right? And notice it's a character. There's also an attribute, but it's going to look at the SNP locations, which is character. So in other words, this is going to be character string, and then it's going to get concatenated with this instance with, and then the length of the objects will be 2. The length of the object SNPs, and then it concatenates with this, and then the genome. So let's execute this and see what we get, and then map it back to that particular function. It says SNP locations. Okay, it got that from the class, right? Instance with 2, which is the length, SNPs on genome H19, which it got from the generic function genome. So in other words, I used a generic function in here to return HD19. So this is just giving you information. It's like a print method. It tells you what the object is. It's a SNP location instance, right? It's an instance of the class SNP locations with 2 SNPs on genome HD19. Okay, so that's sort of equivalent to the print method that we have for normal S3 type objects. Now, one of the nice things is you get this error checking automatically with S4 class instances. You get these automatically. But you can also write your own validity check, and you can associate it with, you can actually put this validity up in the set class method, or we can just define it. We're going to define it here, a set validity, which is all of this, which is going to check. Typically what you do is you check the slots. So when you create it and you give it the arguments for the instance you're going to create, it checks to make sure that you've given it correct information. So HD19 has to be character. The others have to be character vectors of length n, and so forth. And so it will check those types of things. So if you look at the code, it says, okay, we have an object, and for the genome object, it says, is it character? And then the double bars mean or. So is it character? Or is the length of it not equal to 1? In other words, it has to be length 1. It's just one character string. Or is it missing? Is it NA? Is it missing? So in other words, if any of those true, return, then what you return is genome slot must be a single string. That's the message. So if you would type in something that isn't a single string, hopefully it would catch it there. In other words, the first one says if it's not a character, or if it's not of length 1, or if it's missing, then I'm going to print this message. So it has, in other words, what that's saying, it has to be a character string of length 1. So I'm going to execute this, but I'll come back to the slot links in a minute. So we'll execute that. So let's look, now how do we check these slots? Well, what we're going to do is we're going to define the slot length. We're going to take the length of each of those, the length of snip ID, snip chromes. They should all be 2, right? They should all be 2. So I'm going to form a vector which should be 2, 2, 2. Okay, and then I'm going to say, and I say if the length of unique slot lengths is not equal to 1. So what does unique slot lengths do? It takes a vector and it collapses it to the unique values. So in this case, it's going to collapse it what? To a single value of 2. So unique simply does what it says. If I've got 1, 1, 2, 3, you would get a 1, 2, 3 because 1 is repeated. So it's the unique values of a vector. So I have 2, 2, 2 as my vector. If I did it correctly, then I should be 2, 2, 2, or in general, n, n, n. Right? And so it says, so unique in this case would be 2, and it says if the length of the unique slots is not equal to 1, it's a problem. Because they should all be the same, so it should collapse down to a single number. And so if it's not 1, then it prints out the lengths of the slots, snip ID, chrome, and position differ. So you might have 3 of one and 2 of another and 2 of another. So you might put one too many into one of them. And you catch that error also. So what you want to try to catch is up to you. In other words, you're writing code to try to catch input mistakes that people might make. And otherwise, it returns true. So if it didn't return either of those, it returns true. So I executed this, and so set validity. This is snip locations in global environment, and here it gives the name, the genome, snip ID. These are the slots, and character, and integer for the last one. And so if I now run, suppose I assign to Chrome letters 1, 2, 3. In other words, A, B, C, in caps, capital A, capital B, capital C. I've assigned 3 character strings, right? And so this is actually, what this is saying that I'm using the @ sign. I say SNP location at chromosome. I'm assigning it the letters 1, 2, 3. Is that going to work? Well, you can do it, but now let's see if it's a valid object. No, it's not, because now it has length 3. That particular one, in other words, the chromosome has 3. It's a character vector of length 3, and the other 2 have length 2, right? So it says that the slots are not the same lengths, and so it found the error. Okay. Now, what we're going to do next is to find a. Do you remember when I do you remember how I just did this to change the value of the right here in line 76? I use the at sign to change the value for chromosome. So I said, the object at, and then I assign, and that replaced it. But there are also replacement functions where the function is on the right-hand side, and you have your less than hyphen, and then that's a replacement function. That was one of the questions on the exam. So we're going to define a replacement function, and the way we do the replacement function, you notice that this particular right here where it says less than equal, this says for a chromosome I'm going to less than hyphen, I'm going to define a replacement function. And the only thing that I'm going to, initially I'm going to define it as a generic. So chromosome is a generic, standard generic. And except it's not chromosome, it's chromosome, because we've already actually defined that as a standard generic. This is going to be a replacement function, which is going to be a standard generic. Okay? So the difference between this and above is that this has the less than hyphen at the end of it, meaning it's going to allow you to assign to it on the left-hand side. So I'm going to define the generic function. Okay, and then I'm going to do a replacement method. And again, the name of the function, chromosome, and that signature. And then what we're going to do is down here, we're going to say x at chrome gets the value that gets passed as an argument along with x. And then there's a validity check built. This is valid object of x is built right into that method. So that method builds it in. So if I run this, and then now what I'm going to do is I'm going to set this back to the correct. I'm going to say chromosome SNP locations letters 1, 2. In other words, I'm going to make it correct again. So this would make it this will make it correct, but then I'm going to break it again. Let's see what happens when we break it now. But here, notice that I'm using replacement functions. Okay, this is not what I used up here, because I built that into the function right here. Okay, this part right here. So now what happens if I do it? Same error. But so in my replacement function, I actually called the validity check. Okay. Now, another way you might want to do, if I were going to analyze this statistically, I'd like to get this data into a tabular form, right? A data frame. So suppose you had 1,000 SNPs. Well, if I'm going to analyze it statistically, I want it in something like a data frame, right, in a rectangular matrix. So what we're going to do is coerce. We're going to coerce the SNP locations class into a data frame. So notice the second argument is data.frame. So I'm going to, the set as is a coercion type function. Notice it's SAT capital A small s. That's not a mistake. That is the way you write it. And so what I'm going to do, the argument is from. And so in other words, from will be SNP locations. And then I'm going to define a data frame which simply takes the SNP ID equals SNP ID from. See, I've defined these as generic functions, right? So this is going to extract from the object the SNP IDs, the chromosomes. Again, this is a generic function. This is a generic function. So I'm going to coerce this object to a data frame. And so look what happens now. So now, that's the way it prints out in a nice rectangular form, which I can now analyze. So if I were going to do statistical analysis, I typically need to get it into this form. And so I just showed you how to do that. So we actually covered quite a few topics with this example. We didn't cover inheritance. We didn't do that. And in the book, they were talking about prototypes. So when I do my other example, I'll illustrate some other concepts. But this is pretty nice because it went through quite a few ideas, in terms of generic functions and methods. In other words, setting classes, defining methods for those classes, making some of those methods generic functions. You see how convenient they are. Like in the data frame example, it was a very natural way of doing things by using them as generic functions. And so that last one is kind of a nice, this coercion, to a data frame lets us represent the data in a very nice way. Are there any questions on this? Now, I would recommend that you read both that paper. I sort of covered the middle part of it, well, most of the paper. There's sort of a pre-part and a post-part. So you might want to read that whole paper to give you some more background. But again, it's this middle part where we actually form this class that is, I think, important. Now, just remember that a missing step might be, okay, how do we use R to go out to the database and pull data in that would then, we may have to do some further manipulation before we have it in a form that we can create, use the constructor to create the particular object. That would make it more complete. I wonder if the other one's going to work. That is the SNP example. Now, if the other one I'm going to look at, are there any questions on that? I'm sure you have them. You'll have to think about it and read it more than once. Go through the code step by step. Make sure you understand every step of the code. So that gives you, I think it's important to have these types of examples because my feeling is that if you just read the book and I said to do something like this, there's no way that would happen. So with this in mind, Leisch seems to be pretty good about showing you how to do these. He gave a talk. Now, this is actually a little bit old, but so is his other paper. This was written, I guess, in 2004. And again, he presented it as an overview as for classes and methods. His paper that I gave you on LinMod's been particularly, a lot of people will use it as a reference, at least to get started with S3 classes. And this is used by a lot of people to get started with S4 classes. This is a non-genomic example. Oh, well, not exactly. In other words, it actually uses images that are used in heat maps, which are common with two-way clustering, which is very, very common. In other words, if you would have genes and you have subjects and you want to cluster which genes are more similar and you want to cluster which people are most similar, you do it in a matrix. You do a two-way clustering. You get this heat map. And the different colors in the map indicate which genes associated. And if the people are in multiple treatment groups, by looking at the colors, you can see which genes are associated with which treatment groups. So say you have cancer versus not cancer. And then you've got many, many genes, and you're trying to find genomic markers that are associated with a particular cancer. By looking at the color, you can, there are statistical ways, obviously, but this mapping method using heat maps is almost universally used as a graphical representation of what we call two-way clustering, which tells you, can visually tell you which genes are associated with the treatments. In other words, how they differ. In other words, for a given gene, if it's the same for all the subjects in the treatment versus the cancer patients versus the controls, then the color down for that gene will be the same. But if there are differences divided by treatment, we know that gene is associated potentially as a marker for that type of cancer. So that's one of the types of things to do. This is not specifically a cancer example, but this type of thing is widely used in bioinformatics related to cancer. So again, I would take a look at this in more detail, but I don't know if you can see this, but we're going to create something called Pixmap. These are going to be images that we're going to display. And we're going to have this sort of general Pixmap, which has the size of the image, the cell resolution, and the bounding box. So if an image, you have a bounding box. You have a lower left and an upper right, which gives you the bounding box. And then you have the overall size and the resolution. So that's the basic information you have in this class called Pixmap. But then there are actually a couple of ways that we can represent this graphical information. We can do it by RGB. And the other way is an index representation. So we're going to have two subclasses. And in the case, now notice that the two subclasses are going to have the same 3 slots, but the RGB is also going to have something for the red, the green, and the blue to indicate the color. But the colors, in other words, the RGB represents them in terms of numbers for the RG and B, and the combination of those determines the color. Whereas there's another way of doing it using a list of colors with a index matrix. And so that's another way in R that you can represent these images. So these are 2 different ways. And they're both going to inherit from Pixmap, which has the basic information or the basic slots relating to the size and resolution and so forth. Okay? Does that make sense? So this is where we're going to get inheritance. So let's, so again, you might want to read these just to get another person's perspective on, what are classes and what are objects and what are methods and what's inheritance and so forth. Here he discussed what I mentioned before. S is rare because it's both interactive and has a system for object orientation. And so you want the characteristics of functional language, but yet we want object orientation. And as you know, that S3 is a fairly informal method, and it's informal but rather easy, whereas S4 is rather formal but not as easy. Well, of course, maybe if you're working the second assignment, maybe you don't think it's easy, but at any rate, try to mimic sort of some of the stuff that's done in the 2 examples I showed you. At any rate, so he talks about, again, you can read some of this. What I'm going to do is I'm going to start here. Before I do, I just want to mention, he sort of, I know it's too small, so hopefully it's on your screen. Some of the types of things you need is set class, of course, to define classes, and then new to create an instance of a class, and then set generic and set method. We talked about that just last time. So set method sets the methods, and if you want it to be, you also have to associate it with a generic if, in fact, you want it to be a generic function. And then the as and set as to coerce. And then you have things like checking validity, set validity, and valid object are the 2 critical functions, which we used in the last time. And then things like show class, show method, get method. There are lots of these types of things that you can extract information from the object. So, this starts the code, and that's what I'm going to cover. So let's see if this one. I'm going to see if this is going to work. Okay, so if we look at the files, now I'm going down to the Leisch S4 file, and let's bring this up. Okay, and so let's set this as the working directory, and I'm going to clear the console. I'm actually going to clear the graphics. Start out with a clean slate here. So what we're going to first of all do is we're going to define the class Pixmax. Now what are the slots? Well, remember there are 3 slots. The size, which is an integer, the cell resolution, which is numeric, and the bounding box, which is also numeric. Now, the prototype can sort of give initial constructs of this. In this case, size is actually an integer of length 2. Cell resolution is by initial values. In other words, will be 1, 1 initial values, and the BB box will be a numeric of length 4. So let's just create a new one, just based on, so we have the prototype information here, which was discussed in the book. So you can see, by default, there's a print method when you create a new one that says, okay, I have a Pixmap image, and it's a type Pixmap. The size is 0 by 0, and the resolution is 1 by 1, and then the bounding box is 0, 0, 0. Those are just default values that are there. Now I'm going to define the subclasses. So let's look at the RGB one. How does it differ? Well, you already have, this is, in other words, this particular class, Pixmap RGB, is going to inherit from what? That's what this contains is. It inherits from this, the contains equal. And so it has all this. I don't have to repeat the slots for that because it inherits those slots. But I have any new slots for this subclass I have to define. So in fact, what we have to define are the red, green, and blue values. So those are the slots. And the prototype will simply be new Pixmap. In other words, it's simply going to give these same, as if I just created one like this, will be the default values. And we're not giving default values for the red, green, and blue. Okay. And likewise, we're now going to define the other subclass, which is the indexed image. And that, instead of having red, green, and blue, it has the index and the color. And so let's do that. So now if I create a new indexed, suppose I create a new indexed one. So my pick will be of class Pixmap indexed. And so let's see what it looks like. So notice that at this point, the print method was based on that new for the prototype, and therefore it hasn't defined the RGB values at this point. No, it's not RGB, it will be the color and the index for this one. So let's do that. Let's define the index values to be a matrix, a 4x4 matrix of the numbers 1 to 16. So remember, this is saying for the slot index, in other words, my pick at index, for that slot, I'm defining a matrix that will be 1 to 16, and it will be 4x4. And then I'm going to define a heat colors of 16, which, if you look at heat.colors, it actually will partition it, I think, into a 4x4. And then the size will be the dimension of the index. Okay? So now let's look at it. So now, why is that not printing out the... It's still not printing out the slots for the index. But it will once we do a show method. In other words, the default method based upon that new when we did the constructor only printed the basic one. But let's do a show method so that it does print everything out that we need. I'm going to run this, but I'm going to go back and say, okay, let's take a look at the code. Okay, so I'm going to have a show method for Pixmap. Okay, now the show method for Pixmap is to print it out will be the type, will be the class of the object, then the size will be, I'm looking at object size, and then collapse equals x, collapse it into, I'm trying to remember how. I forget exactly what. Let's look it up. That's not it. I don't know if it's this virtual method. Let's take a look at. I'm forgetting how this collapse is defined. So at any rate, what we're doing is we're again looking at size, resolution, and the box bounding length. So we're creating our own method. We're creating our own set method. Did I do that? Yeah. So that's the show method. Again, I'm using a cat, so it's concatenate this to the, in this case, to the class of the object, and then the /n means a new line. And then I'm pasting here, I'm looking at the size. Oh, I think the collapse equals x if it looks at the size. Oh, yeah. It's like 0, x, 0. So if it was a vector 0, 0, it will put an x between them. So when it collapses, if it takes a vector and collapses it, it puts in, the x is the symbol it uses. Now, we're going to have a plot method. We're going to have a plot method that it will take x and y. It's going to have, it's not going to, x lab is there are no labels for the x and y axes. And in fact, we're not going to use axes. We're just going to have a heat map with an image with no x or y axis and no labels. So we have blanks for that, and the axes are false. The aspect ratio is 1, meaning the horizontal to the vertical length rise is a 1-to-1 relationship. So those are defaults, dot xxx. So there are other arguments to these plotting functions. Plot is a generic function. You've used it before. If I say plot xy, well, what it plots depends upon, what x and y are. So if the x and y are both numeric, it does a scatter plot, for example. So in this case, you could pass other graphing parameters to this. But in this case, whether or not it uses it. So there's a function in R called image, which plots these images. And these are high-resolution images. And a lot of times if you publish in journal articles, you'll use this function image, because it does very nice high-resolution image. So technically, you could take a photo and pull it in. And you could pull it in, and you could actually use R to manipulate that image. One of the things that people are confused about is they think that analyzing data, a lot of people think that we mainly analyze numeric data. And that's actually a big distinction between what mathematicians do and what statisticians typically do, is that part of our data is numeric. So we may measure a lot of numeric data. But if you think about functions in mathematics, y equals f of x, so we're talking about numeric x and y typically. But we have categorical variables, and we have ordered categorical variables. So we analyze categorical information as well as numeric information. But increasingly, the type of information we analyze is not not just categorical values or numerical data. It's also things like images. You may analyze documents. So the objects you analyze could be documents, they could be images, they could be web pages, et cetera, et cetera. So that as you move into what's often called data science, you actually are analyzing far more. So if you took an image, for example, and you were doing character recognition, or you were doing, so you have some characters in it, or you have a person walks through a scanner and there's a snapshot of them, and then you immediately want to match that picture to this database, say in an airport. Then that involves algorithms on the image itself. At any rate, so we have so we're going to, I don't particularly know this particular image very well, but this is, you're passing it the size and so forth, the color information, the size, the index info. The index is a matrix, and so you're looking at the information relating to the size for the index as well as the color. And then notice that ... if you passed other graphing parameters here, they would also be passed to image. So if you look at the options that image would take, in other words, the graphics options, you could pass this through to that. So this is the actual thing that does the plotting. So let's now do a new Pixmap, and now let's do my pick, which we had defined before, and let's do the basic plot to my pick, and this is what you get. Notice it's 4 by 4, and this is sort of the color mapping. Now, as I said, this we're just using a scale, starting, it's getting lighter red, and it's scaling down. That's the sort of default values we had defined for this image map. But in reality, if you did a genomics experiment and you had a heat map, then you could actually functionally define what those colors are. In other words, this color might represent a particular gene in a particular person. So if these are people and these are genes, the color map for that would represent a particular, for every gene of that, well, not every gene necessarily, but if you're looking at microarray expressions, for whatever genes, and it might be thousands for every person. So you could have thousands of columns, and these types of problems, you actually have far more genes usually than you have people. You may only have 100 people, so you may have 100 rows and 1,000 columns. Of course, you wouldn't look at that whole thing. You would probably look at a subset of the genes, not all of them, because it might be kind of hard to plot something with 1,000. I mean, you'd have to have the column widths very thin to do that. Let's put it that way. So at any rate, that's sort of it's a basic image. Now, what are the slot names for my pick? So the slot names are index, color, size, cell resolution, and inbox. Those are the slot names. And now, suppose we want to coerce my pick into a Pixmap. So what this will do is it will take my pick and coerce it into a pick map. Right now it's an indexed object, and I'm going to coerce it, and I'm going to call it my pick 1. So now if I look, since it's a pick map, you don't have the slots for the index and the color. So since I coerce it, I only have these slots, because I now coerce that object into a basic pick map and no longer an index plot. Now, that presumably means that you're not going to be able to graph it. Now, suppose I create a new Pixmap. I'm going to create a new Pixmap, and I'm going to use the characteristics of the Pixmap 1. So I'm going to have this Pixmap 2 created. And so what I did is I'm creating a new object of Pixmap RGB based upon this Pixmap 1, which is actually a basic Pixmap. So If I look at it though, I now have all the slots, including the RGB. So I created a new Pixmap from the basic Pixmap RGB from Pixmap 1, which is just a Pixmap, which doesn't have the extra slots. Okay. Now, suppose I have, in order to use that image plot, I had to have it indexed, right? So suppose I have an RGB and I want to use the image, but the image is requiring that I have the index type plot. So if I get an image, say I took a picture and I had this image in RGB, then I would have to convert it to this other format, right? So what I'm going to do here is I'm going to I'm going to coerce the Pixmap type image to an index type. And so I have a from to. And so what I'm going to do is I have a function from to, and I'm going to take the RGB function, and I take the red, the green, I'm taking the red, the green, and the blue, and assigning that to X, and then I find the unique values of that, and then I'm going to match these RGB colors to the colors in the Pixmap, and let's see. Okay, these find the unique colors from this RGB, and I'm going to find the matching ones, and then I'm going to create a matrix which depends upon the number of rows from the from. The two subscripts, the first and the second, give me the rows and columns. And so at this point now, I'm going to create a new Pixmap image. What time are we getting today? Okay, I guess I've gone over. So at any rate, if I do this, I'll stop in just a second. Okay, and I'm going to quickly do this, but I'll come back to this next time. She has a class. She's teaching. So let's see. Let me just quickly do this, and then I'll talk about it more next time. I'm going to go through these quickly, and then I'm going to create and this was the way we transformed it, by using sines and cosines. And so this is what it now looks like. And we did a filtering process based upon sines and cosines of that original.
Is that a heat map?
Well, it's not exactly a heat map. It's a filtering technique. It's a mathematical filtering technique that's based on sines and cosines. Okay, I'll talk about this a little bit more next time.
Is that first thing more of a heat map?
That's a heat map.
That's a heat map?
I could show you some examples. It's very common in bioinformatics papers to see these heat maps. And usually the columns may represent genes. Well, a lot of times the rows represent a gene and the columns represent. It depends. Depends on how many subjects you have. And usually,
I think I've seen it before.
Yeah, I mean, typically you may have the subjects across the top and the genes down. But it doesn't have to be genes. It could be proteins. It could be, it could be any types of things you measure on each person. And you do a two-way clustering, and then based upon the values, you assign different colors. And you're looking for patterns.
Good stuff. I like the biology integration.
Yeah, I'm going to be doing more of that as we go along.